All living things are composed of one or more cells. All cells come from pre-existing cells, but not all cells are made the same. However, all cells do share certain characteristics in common. So let's start with the characteristics that all cells share. All cells will have these features. They will have a plasma membrane and cytosol. Now that plasma membrane is the exterior portion of the cell. It's the outside. It's the border of the cell, the skin of the cell, if that helps you think about it. If you remember at the end of Unit 1, I talked about a phospholipid bilayer. You remember phospholipids, that kind of oily sheet membrane, and I drew them coming around into a circle, creating this kind of oily membrane. The phospholipid bilayer, the plasma membrane, is that oily sheet. So it's this kind of liquid membrane that surrounds all cells. And that's everything. Plant cells have them, animal cells have them, fungus cells have them, bacterial cells have them. All cells have that plasma membrane. The cytosol is the interior fluid of the cell. It's this mix of water and proteins and sugars and amino acids and salts and just everything that the cell might need at any given time, all floating around mixed together. And you might imagine it has a degree of viscosity to it. Sometimes it's called a jelly-like fluid or a jam-like fluid inside the cell. I think it's more helpful to think of it as honey-like myself, because at least honey has a little bit of flow to it, unlike most jellies that I've seen, but it's a viscous fluid is the important takeaway there. So all cells will have the plasma membrane and the cytosol. Oh, incidentally, I should probably mention this right away. The cytosol is the fluid inside of the cell. The cytoplasm is the fluid and all of its contents, including the organelles and whatnot. So sometimes you'll see those two terms, cytosol and cytoplasm, used almost interchangeably. That's why, because they are usually referring to approximately the same space. The cytoplasm is essentially everything between the nucleus, the location where all the DNA is, and the outer membrane, the plasma membrane, whereas the cytosol is specifically the fluid that's inside the cell. The DNA will be another category that all cells are going to have in common. So not only do all cells have these membranes and the cytosol, they also all have DNA. And not only do they all use DNA as their genetic material, they use the same DNA code. They use the same 4 DNA bases, A, T, C, and G. We'll talk more about that later on. And they read the code in exactly the same way. And there's no reason why they should have to do that. AUG means methionine to your cells. AUG means methionine to these Streptococcus bacteria as well. All life will read the same series of letters, DNA letters, in exactly the same way, even though the code is more or less arbitrary. So that's a really interesting thing that all life on Earth has in common, that they share this chemical language. Another thing that all living cells have in common are ribosomes. So regardless of whether they're talking about human cells or fungus cells or bacteria cells, all cells have to take information from their DNA, from their genes, and make proteins out of it. Proteins, again, are what gets all of the work done inside of your cells, so you need to build them, right? The DNA just holds the instructions for building the proteins. The proteins do all the work. How do you get the information into the actual proteins? Well, you need ribosomes to build those proteins. So these ribosomes are these tiny little structures that put together the correct amino acids in the correct order in order to make sure that your proteins are functioning properly. So these 3 features are things that all cells, regardless of which domain or kingdom of life a cell comes from, are going to have in common. But I'm getting ahead of myself talking about domains and kingdoms. Let's start even broader than that. All organisms on the planet can be placed into one of 2 categories. These are prokaryotic organisms and eukaryotic organisms. Prokaryotic organisms are single-celled, and those cells are very small, teeny tiny little cells, and they're very primitive by the standards of, say, our cells. So bacteria are prokaryotic cells. they're not the only kind of prokaryotic cells. There's also another category which we refer to as Archaea, which frankly get a little bit short shrift in this class. We don't discuss Archaea too much. And that is unfortunate because Archaea are extremely fascinating organisms. One, they are often extremophiles living in boiling water, like hydrothermal vents where there's boiling water, or extremely salty or sulfury conditions. Two, they have linear DNA like we have linear DNA. They have a lot of features in common with our cells that bacteria would lack, so that's an interesting little feature there. But when you think prokaryotic cells, usually it's pretty easy to just kind of say, oh, prokaryotic, that's the bacteria one in this class, and you'll be correct on that count. The other type is eukaryotic cells, and eukaryotic cells are the organisms that you are generally more familiar with. Plants are eukaryotic organisms. Funguses are eukaryotic organisms. Animals are eukaryotic organisms. Therefore, we are eukaryotic organisms. And protists are eukaryotic organisms. Protists include things like algae and these sorts of things. Now, within that prokaryotic category, Archaea and bacteria are what we refer to as domains. So there are two prokaryotic domains of life. There is one eukaryotic domain of life. So we have 3 domains total. You can put all living organisms into one big category called life, and then the next division down is these 3 domains: Archaea, bacteria, Eukarya. And then if we were to subdivide Eukarya down, we would then cut it up into 4 kingdoms. And here are 4 kingdoms of domain Eukarya. We have kingdom Plantae, kingdom Animalia, kingdom Fungi, and kingdom Protista. Now, what defines each one of these kingdoms? Well, part of it comes down to what features their cells have, and part of it comes down to their feeding behavior. So, kingdom Plantae, you have the plants, and you guys already know a little bit about their cells. These are going to be photosynthetic organisms, usually, so they will have chloroplasts in their cells. They'll be able to do photosynthesis. They're what we call autotrophs, self-feeders. Autotroph, auto means self, troph means feeders. So they're able to do photosynthesis, get their energy from sunlight, and their cells are surrounded with a cell wall. That's a really good key giveaway that it's a plant. Another good key giveaway is that it's a multicellular organism. Plants are multicellular by definition, as are animals. Animals are multicellular by definition. So here we have some animals. I know that they're animals because they're multicellular. Their cells do not have any cell walls. If I were to take a sample, they would not have any cell walls around them. And they are heterotrophs, and heterotroph means other feeders. So they need to feed on other types of organisms in order to meet their energy needs. Now, that doesn't necessarily mean being a carnivore. It could mean being an herbivore or a fungivore or any other kind of vor that you like, but being an animal, you do need to eat some other kind of organic matter in order to meet your energy requirements. Now, fungi are also a kind of heterotroph, an other feeder, like animals are. In fact, fungi are kind of similar to animals in a lot of ways, but animals have a kind of internal digestion system, right? We take food into us in order to digest it. Whereas fungi typically just grow on top of whatever it is that they are eating. They extend little fibers of themselves into whatever it is that they are growing on top of, which secrete enzymes, and those enzymes help break it down. And as it breaks down into its component parts, they're able to absorb those nutrients in order to help build themselves up. But in that process of breaking it down, they are decomposing it, which is why we call them decomposers. So fungi are heterotrophs. Fungi, like the plants, have cell walls around their cells. So they're like the plants in that they have cell walls. They are like the animals in that they are heterotrophs. They can't do photosynthesis. You'll never find chlorophyll inside of a fungi. One big difference between fungi and plants or animals, though, is that they can be single-celled or multicellular. You have, for example, these mushrooms here. These are multicellular fungi, but brewer's yeast or bread yeast, like you would use for baking a loaf of bread, that is a single-celled fungus. So plants and animals, multicellular. Fungi could be multicellular or single-cellular. What about these guys down here at the bottom? I haven't mentioned anything about them. What about this Kingdom Protista situation down here? Kingdom Protista is kind of complicated. Okay, can I be real with you guys about Kingdom Protista? It's really about 36 different kingdoms that we've just sort of tucked into one big kingdom because we didn't want to sort it out. I'm kidding. Kind of. It's not so much that it contains so many kingdoms and we didn't want to sort it out. In the process of sorting it out and figuring out the cladistics of all of the different groups involved, we've realized that some organisms that we call protists are actually more closely related to animals than they are to other protists, while others are actually more related to plants than they are to other protists, and that there was no neat, organized way for us to say that, hey, all of these organisms are more related to each other than they are to the others, and sort them out in such a way that wouldn't essentially break our entire classification system. So nowadays, the term protist isn't so much representative of a single closely related group of organisms as it is the term of convenience that we use to describe all eukaryotic organisms, all organisms in the domain Eukarya that are not plants, animals, and fungi. Sometimes it's referred to as the junk drawer of Eukarya. So if that helps you think about it, then that's fine. And the reason that that might be helpful is because you get lots of different traits, lots of diversity in Kingdom Protista. So you could get organisms that are single-celled, like these Euglena here, or you can get multicellular protists, like giant kelp. Seaweed is actually a kind of algae rather than being a kind of plant. You can get protists that are able to do photosynthesis, like that algae, and like these Euglena, but these Euglena can also hunt for their own food. They're heterotrophs, like fungi and animals are. They're actually able to swim around and grab other organisms and swallow them for nutrition as well. And then you have other protists that are able to just use hunting and feeding in order to meet their nutritional requirements. When you get protists that behave a lot like animals, we usually call them protozoa. When you get protists that seem a lot more like plants, we usually refer to them as algae. When you get ones that are a little bit more like funguses, sometimes we call them slime molds. So you really get a lot of diversity in Kingdom Protista. So all 4 of these kingdoms are eukaryotic, which means that they have large, complex cells. Now, that term, eukaryotic, means true nucleus, that they have a nucleus. We're going to talk about cellular organelles in just a second, and the nucleus is one of the big ones. The nucleus is the container for DNA inside your cells. Eukaryotes have a nucleus, but bacteria and archaea do not have a nucleus. They do have DNA, but they don't have that container. So plants, animals, fungi, and protists, they're eukaryotes because they have a nucleus, whereas, next slide, the prokaryotes do not have a nucleus. So up here at the top of the slide, we have some bacteria, a kind called Pseudomonas aeruginosa. Notice the 2-part name. We'll talk about that more when we talk about evolution and how scientific names work. You always get these 2-part scientific names for a species. So this is Pseudomonas aeruginosa, a kind of bacteria, and bacteria are one type of prokaryote. What makes a prokaryote a prokaryote? Well, they are small cells. Like we saw on that size scale, they are 1/100th to 1/1000th, sometimes even 1/10,000th the size of a eukaryotic cell. They lack internal structures which are surrounded by their own membranes. This part sometimes confuses people. All cells have a plasma membrane. They have the outer membrane, the exterior membrane, the outside one. But prokaryotes do not have internal compartments surrounded by their own membranes, whereas eukaryotes have lots of different components inside their cells, each of which have their own membranes. Prokaryotes don't have any of those. So like the nucleus, for example. And the next item on our list is that obviously, if they don't have any of those, and the nucleus is one of them, that prokaryotic cells do not have a nucleus. In fact, the name prokaryote means before the nucleus. Karyot comes from the Greek word for kernel or seed, so when we were looking at cells under the microscope, we saw that some of them had this little dot in them. If you remember looking at the skin cells, that little blue dot for the nucleus, we called that the kernel or the seed, the nucleus, the karyot. And we noticed that some cells had it and some cells didn't. We kind of assumed that these bacteria were more primitive, so we said these are the ones that came before the nucleus, the prokaryotes, the before the nucleus cells. So lacking a nucleus is one of their defining features. So small, no internal membranes, no membrane-bound organelles is the way I'll usually say that. They do not have any membrane-bound organelles, and they do not have a nucleus. They have a cell wall, though. So like plants and like fungi, they will have a cell wall. But plants have a cell wall made out of cellulose. Fungi have a cell wall made out of a material called chitin. And bacteria have a cell wall made out of a material called peptidoglycan, which the name peptidoglycan might give you some intuition that there's a mixture of amino acids and sugars being used there. We're not going to go over the chemical structure, but it's a different material than the plant cell wall. So that's why I mention it here, is that yes, they have a cell wall, but it is a different kind of cell wall than a plant cell wall. And they're very, very old, the prokaryotes. Bacteria and archaea originated about 3.5 billion years ago. They have been on this planet for a very, very long time. We are living in a microbial world. This is very much their planet, and we are just occupying it for the time being. One of the things that every biology student, no matter what level you're at, has to be able to do is take a look at a cell and diagram its parts. So let's take a look at this diagram. Now you'll notice around the exterior of this Pseudomonas aeruginosa bacterial cell, you actually have 3 layers. You don't just have the plasma membrane. We've already mentioned the plasma membrane, and that's this yellow layer here. But we also have the cell wall, and that's outside of the plasma membrane. So you have 2 layers here. You have the fluid plasma membrane, and then you have the suit of armor, the cell wall, which provides a little bit of rigidity to that cell. The purpose of the plasma membrane is to control what comes in and out of the cell. The cell wall, on the other hand, provides structure and protection for that cell. But on the outside, we have what's called the capsule. This is called the sugar capsule. It's also sometimes called the glycocalyx, which I'm a big fan of because I like the fancy words, the glycocalyx, but you can just call it the capsule. It is a sugary secretion of the cell, which is very, very sticky, and because it's sticky, it allows the bacteria to adhere to surfaces, which is very handy if you need to, for example, attach yourself to the inside of someone's digestive system or into nooks and crannies in their skin or on a doorknob or something like that. If you need to adhere yourself to a surface, it helps to have a little bit of stickiness to you, particularly if you're in an environment like the small intestine, where there is a constant flow of material trying to dislodge you. It helps to be able to attach yourself very strongly. So the capsule helps with that. This is also where a lot of the toxins that bacteria secrete and make us sick might be found inside of this capsule coating. Now, coming off of this capsule, we have, it looks like this bacteria needs a shave, right? They got all these little hairs coming off of it. These are called Fimbriae. And what these do is increase the surface area. And, oh God, surface area. It seems like half the answers in biology are all about surface area, aren't they? Because these Fimbriae increase the surface area, they make it even stronger of an attachment to other kinds of surfaces. So they help the bacteria to stick to surfaces even more than the capsule alone would have allowed. So that covers the outside of this prokaryotic cell, the plasma membrane, the cell wall, the sugar capsule, and the Fimbriae. So let's take a look. Let's before we move inside, let's take a look at this back end here, we have the flagellum. You've probably seen flagellum before. You saw some in health class, probably when you were a good deal younger. Sperm, right? Sperm and egg. You've seen those little videos of fertilization occurring. Sperm cells have flagella as well, these little whip-like structures that propel cells forward. Now, prokaryotic cells are very different from eukaryotic cells. Sperm, obviously, they're our cells, they're eukaryotic. So these are structurally very different. Prokaryotic flagella also are built differently from the ones that would be on our sperm cells. For example, these are made out of a single long protein filament, and it spins around 360 degrees. And by spinning around, it actually creates a little propeller, like a motor, like a speedboat motor, which causes the bacteria to be able to cruise forward. If it spins one direction, it takes it into a kind of tumbling motion. If it spins another direction, it sort of pushes it forward with the current. And through some combination of tumbling and moving forward, it's able to redirect itself and find its way around its environment. So these flagella are for propulsion. It's for helping it swim its way through the environment, and it's very common to see single-celled organisms with more than one flagellum. The word flagellum, incidentally, means whip. It means whip-like. So this is a whip-like method of propulsion. So we have the flagella, the plasma membrane, cell wall, capsule, Fimbriae. Let's move inside of this bacterium. We have the cytosol, which is all of this light blue area. It's that fluid inside, and floating around in this cytosol, we have these little dots. These dots are the ribosomes. Ribosomes are what we call an organelle, a tiny organ that floats around inside of the cytosol. Now, prokaryotic cells don't have any organelles which have their own membranes, but ribosomes don't have their own membranes. So they're allowed to have ribosomes, and that's one of the only organelles that you'll find inside of a prokaryotic cell. Ribosomes help make protein. They take information from DNA, and then they translate that information into proteins. And since all living things need protein in order to function, well, all living things are going to need ribosomes as well. So you'll find ribosomes here. It's one of those features that all cells have in common. And then you find DNA. So bacteria also use DNA just like we use DNA, but no nucleus. Because remember, prokaryote, before the nucleus, before we had nuclei in cells. So they have DNA, but they do not have a membrane-bound compartment, no box to keep that DNA inside of. Instead, they just collect all of the DNA into a big old lump called the nucleoid region, kind of a pile of laundry approach to storing your DNA. The DNA for bacteria is similar to our DNA in that it uses the same 4 bases, A, T, C, G, and it reads the code the same way we do. It's dissimilar in an important way, though. Our DNA is linear. Our chromosomes could be unraveled to form straight lines, whereas bacterial DNA is circular. If you were to unwrap a bacterial chromosome, it would make one big circular, like an Ouroboros, like it would wrap back onto itself, making a big old circular thing, which is interesting. So they have DNA, just like we have DNA, although they store it differently. We have a nucleus, they have a nucleoid region. So if I were to give you this diagram on an exam, you should be able to tell me what any given one of these portions are. Now, if this diagram is overwhelming, I have some bad news, because prokaryotic cells are very primitive, especially when compared to eukaryotic cells. And in the next video, we're going to start a tour of eukaryotic cells, and there are a lot more labels there.